Today I'm going to discuss the method with which we could determine the 3D structure of a protein using solid-state NMR. So as you know, proteins are made of amino acids and they are primarily available as sequences. These sequences fold into certain shapes to have secondary structure, and then they again fold spatially to have a tertiary structure. And if these structures interact with, if there are like interact with each other, or let's say if they have identical subunits, then they form the quaternary structure of the protein. So using solid state NMR spectroscopy, first we do a lot of experiments like spin diffusion experiments, CHHC, NHHC, to assign or identify the amino acids. Then we probe in this same kind of experiments for the short range contacts, like one amino acid talking to the next one or what to the one previous, and this will give information about the secondary structure, whether this is an alpha helic conformation or beta strand conformation. And we again do this set of experiments with a lot of mixing times, like we allow the nuclei to talk to the distant ones. So we mix higher, then the longer the distance is, amino acids can talk, meaning the nuclei can talk, and with this information we could generate the tertiary structure. Usually we get distance information, and with the chemical shift assignments of the nuclei we could get the dihedral angle information as well. And with, if we are lucky, then we could also get the intermolecular contacts with which we could calculate the quaternary structure. One slide which I want to really show you is that is this one. Now this is comparison of a solid state NMR spectroscopy with a solution state NMR spectroscopy. In solution state NMR we don't have this in this, if you consider this as a molecules, they just move here and there, because of the Brownian motion, and you get really nice spectra. But in solid state NMR we don't have this. We have this in static mode as shown in the picture. The resultant is we have really strong interactions, anisotropic interactions playing in, making our spectra broad compared to this sharp line of the solution state NMR. But we definitely can get this kind of spectra in solid state NMR using a technique called magic angle spinning, which I will discuss in the next lecture.